Hi, my name is Huldah, and this is my project titled Bugging the Infant Brain with Bifidobacterium, the Role of Bifidobacteria in Infant Neurodevelopmental Outcomes. It is often said that children develop at their own pace, but what if I told you that a bacteria called Bifidobacterium actually sets that pace? Bifidobacterium is a bacteria that is highly abundant in the infant microbiome during critical windows for neurodevelopment, and is considered a key species for infants in the first 1,000 days of life. Previous literature suggests that bifidos may play a helpful role in early neurodevelopment, especially for fine motor skills. By neurodevelopment, I'm referring to the maturation of the central nervous system underlying the developmental outcomes of children. The question you're probably thinking about right now is, how does the bacteria in the gut influence neuroprocessing in the brain? That would be through the gut-brain axis. The gut-brain axis describes the continual back and forth communication between the brain and the gut. In the systemic pathway of communication, metabolites travel through the bloodstream and interact with neurons. While bifidos themselves can't cross the blood-brain barrier, their derived metabolites can. But how does that implicate future development? DOHAD is a well-supported theory stating that an imbalance of beneficial bacteria during sensitive phases of development is strongly predictive of poor health and developmental outcomes. In particular, 3 months is identified as a really important time period for acquiring healthy gut microbiota. To date, no study has explored bifidobacterium and its derived metabolites at 3 months. So, why is this important? 1 in 4 Canadian children are at risk for developmental delay or disability. Understanding the role of bifidobacterium can propel microbiome based therapies to support the development of children that are at risk. So, that takes us to my project. In a nutshell, my project's objective was to clarify this relationship by investigating the correlation between the relative abundance of bifidos at 3 months and neurodevelopment scores at 1 year. Data for this study came from the pregnancy during the COVID-19 pandemic cohort, a sample of 878 pregnant mothers and their kids. We selected 8 covariates with strong support from the literature to control for potential confounding in our model. For our exposure variable, infant stool samples were collected at 3 months, and the microbiome was analyzed using metagenomic shotgun sequencing to identify bifido species and mass spectroscopy to explore the metabolic activity in the gut. For our outcome variable, parents completed the ASQ3 questionnaire, a screening tool that assesses neurodevelopment across 5 domains. The ASQ3 provides categorical cutoff scores with information on whether a child is developing typically or at risk, and continuous overall domain scores. So we used continuous data for the present study. Lastly, for our analysis, we ran a linear regression model and used the Benjamini-Hochberg method for FDR correction. Our significance level was 0.05 with a 95% confidence interval. So with that said, let's jump into the results. So this heat map shows the species that are present in more than 10% of the infants in our study. The reddest patches in each section are the participants with the highest abundance of that species. So for example, Bifidobacterium longum in the first column is visually the reddest species, and that's because it's present in 82.2% of our sample. So we ran a regression analysis between the 6 dominant bifido species and ASQ3 domain scores and found both a positive and negative trend in our downstream analysis. Bifidobacterium pseudocantinulatum had a positive association with fine motor and problem-solving domains at 1 year, where higher abundance predicted better performance. Higher abundance of Bifidobacterium bifidum and Bifidobacterium adolescentis predicted worse performance across all domains, except for gross motor domain, which resulted in better performance. So none of our results survived FTR correction for multiple comparisons. In conclusion, our results align with findings on key infant-type bifido species and the dominant effect of bifidos on fine motor skills. Bifidos may also have specific roles for development. While none of our results remain significant after FDR, they are still worth mentioning. There are trends in our data which suggest that an obscure but strong association does exist, which warrants future research. Future directions can include running a logistic regression analysis to assess clinical significance, assessing the effect of metabolites on ASQ3 scores, and probing the association between bifidobacterium, metabolites, and neurodevelopmental outcomes. And that concludes my presentation. Thank you so very much for your attention.